Welcome to Bio Bits and Bytes. Think about how fast things are changing in science and tech these days. It's pretty incredible, right?
It really is almost dizzying sometimes.
Yeah, and today we're diving into this really exciting point where two massive forces are meeting. We're talking artificial intelligence, AI, and our understanding of biology right down at the code level genomics.
And how putting those two together is starting to, well, crack open one of the biggest nuts we've ever tried to crack, finding new medicines.
Exactly. So this deep dive, it's all about how this cutting-edge AI is changing the game for drug discovery. We're looking at how it's trained on these huge, sometimes really unique biological data sets.
Right, data sets that go way beyond what's just publicly available.
And we'll focus a bit on finding drugs from nature, natural products, and also designing completely new genetic medicines.
And we got some great source material to guide us today. There's stuff from a company right in the middle of this. Basecamp Research funding news, strategic moves, gives us a real-world picture. Plus, we're drawing on some solid scientific literature. A review paper on deep learning's role in drug design, and another one digging into environmental DNA, or eDNA, and how that unlocks natural products from microbes.
Okay, so our mission today really is to unpack all of this, pull out the key insights, connect the dots.
Show how it all fits together, the AI, the genomics, the biology from nature.
Right, and how it could lead to maybe some truly transformative treatments. But also, let's be real about the challenges because it's not all smooth sailing.
Definitely not. There are big hurdles still. We're genuinely exploring beyond known biology, as Basecamp puts it. It's exciting stuff.
So glad you could join us for this exploration. Let's jump right in with Basecamp Research. They're making waves because they sit right at that intersection, AI and life sciences.
Yeah, and their mission statement, like you said, to solve pressing challenges by exploring beyond known biology. It really catches your attention.
It does. It signals they're not just iterating, they're trying to leapfrog.
And a key part of their strategy, according to the sources, is building their own AI platforms. But the crucial bit, they're training them on proprietary data.
Proprietary meaning stuff only they have access to, not the public database.
Exactly. Data they collect themselves, which they believe gives their AI an edge because it sees biological information that others simply don't. They argue this is essential for designing really sophisticated, novel biological solutions.
And it looks like investors are buying into that argument. Big time. That recent funding round.
Oh yeah, the $60 million Series B, that's significant, led by Singular.
And that brings their total funding up to, what, $85 million now?
That's right, $85 million total. And look who's involved. It's not just the usual VC firms like Singular, S32, Reed Alpine.
Right, the source mentioned individuals too. People
Yeah.
from big pharma, consumer goods, Roche, Philips, Unilever. That's interesting.
It really is. It shows confidence from both the tech side, who get the AI, and the life science side, who tightly understand the need for new drugs and the potential market. It's not just a tech bet.
It's a biology bet, fueled by tech. And the source is pretty specific about where that money's going, isn't it? A lot of it is aimed squarely at scaling up that data collection.
Absolutely. Ramping up the pace and the sheer volume of gathering that unique proprietary data, they're doubling down on building out their foundational data set.
There was that quote that really stood out. Their data set having 100 times more advanced biological systems than the public databases most used by pharma researchers. 100 times.
Yeah, and think about what that means. If everyone else is training their AI on the same public stuff, they'll likely find similar things.
Right, they're all fishing in the same pond.
Basecamp is essentially saying they've built their own much, much larger and maybe differently structured pond, so their AI might spot things, patterns, connections that are just invisible in the public data, entirely new insights.
And they're collecting it incredibly fast now. The source said 10 times more data gathered in just the 3 months before that article came out compared to the whole 3 years before that.
That's exponential growth. It points to a really sophisticated, globally scaled collection operation they've put in place.
So more unique data, faster. And the funding also beefs up the other side, the AI itself.
Yes, strengthening the AI capabilities, which includes, crucially, investing heavily in the compute power, the actual hardware needed. They built one of the most advanced industrial computing infrastructures in Europe.
And they mentioned the specific chips, NVIDIA H200 GPUs. Why does that detail matter? Is it just about speed?
Speed is part of it, but it's more about the type of computation they enable. H200s are, top tier. But the key thing the source highlights is that this setup is purpose-built for training long-context AI foundation models.
Long context. Okay, Break that down for us. Why is long context so important for biology?
Sure. Think about biological data. A gene, a protein sequence, a whole genome. These could be incredibly long strings of information.
Like a really, really long sentence or paragraph.
Exactly. And the meaning, or the function, often depends on relationships between parts that are very far apart in that sequence. Standard AI might struggle to keep track of those long-range dependencies. These long context models running on hardware like H200s are specifically designed to process and understand those connections across vast stretches of sequence data. That's vital for genomics.
Got it. So the hardware is tailored to the unique nature of biological data. And where is this unique data actually coming from? You mentioned a global operation.
Yeah, they describe it as one of the world's largest commercial biodiscovery programs. It's not just one lab bench. They're operating in over 120 locations across more than 25 countries.
Wow, why so widespread?
Diversity. Biological solutions aren't uniform. What works in one environment, or the organisms found there, might be completely different from another. Think rainforest soil versus a deep-sea thermal vent.
Totally different worlds.
Right. So by sampling globally, they aim to give their AI a truly comprehensive view of biology's breadth and complexity. Capturing that natural variation is key. And the source also makes a point of mentioning the data is ethically sourced, which is obviously critical.
Absolutely essential. Okay, so massive unique data, powerful tailored compute, but they're also investing in people and location, right? The Boston move.
Yes, setting up a new lab and office in Kendall Square, Boston. That's like ground zero for biotech,
The heart of the action.
pretty much. And the source says this move is specifically to accelerate its pursuit of programmable genetic medicines. Being there helps tap into talent, collaborations, the whole ecosystem.
And speaking of talent, they brought in a new chief scientific officer, John Finn. His background seems like a perfect match.
It really does. The source calls him a globally recognized leader in cell and gene therapy, with over 25 years tackling really tough scientific problems.
And his previous role at Tome Biosciences, pioneering large gene insertion, that sounds directly relevant to genetic medicines.
Highly relevant. That's one of the big technical challenges, getting large pieces of genetic code inserted correctly. He also has experience at other key players like Kodiak and Intellia. He knows this space inside out.
And his quote in the source suggests he was drawn specifically by Basecamp's unique approach.
Exactly. He said he saw the opportunity to develop the next generation of programmable genetic medicines, and that Basecamp's vast and unique data set, combined with its next generation of AI models, was highly complementary to his own expertise. It suggests a real synergy.
Like the platform is enabling the kind of science he wants to do.
Precisely. And they're already putting this combination to work. They have partnerships, like that big multi-year one with the Liu Lab at the Broad Institute.
David Liu's lab, wow, he's a giant in gene editing.
He is. And collaborations with, I think it was 3 of the top 10 pharmaceutical companies. So big pharma is clearly seeing value in their platform, too.
Okay, so Basecamp is a compelling example of this convergence. Unique data, powerful AI, expert talent, all focused on breaking new ground, especially in genetic medicine. But this leads us to the core question. What is the AI actually doing? How does this deep learning stuff work for designing drugs? Let's dig into that review paper.
Sounds good. And maybe it's worth quickly defining those key terms first. AI, ML, DL, just to make sure we're all grounded.
Yeah, good idea. Let's start with the big one, AI.
Okay, artificial intelligence, AI, is the broadest term. It's basically about building computer systems that can do things that normally require human intelligence. Thinking, learning, problem-solving, understanding language,
Yeah.
simulating intelligence.
The umbrella term added.
Underneath that, you have machine learning, ML. This is a subset of AI where the system learns from data. Instead of programming explicit rules for every situation, you feed it examples and learns patterns and how to make predictions or decisions based on that data.
Learning by example, essentially.
Exactly. And then we get to deep learning, DL, which is a specific type of machine learning. The deep part refers to the structure. It uses multiple layers of interconnected processing units, kind of like neurons in a brain.
Multiple layers, like processing information step by step, getting more complex.
That's the idea. Each layer learns increasingly abstract features from the data passed on by the layer below. This depth allows DR models to tackle incredibly complex patterns, the kind found in images, language, and definitely in biological data. The source notes DL really pushed AI forward in many fields.
And it's particularly suited for drug discovery because biological data is so complex.
Precisely. The review paper really stresses the need to boost the use of DL in pharma R&D, saying ML and DL have clearly enhanced drug discovery and design. They can model these intricate biological systems in ways older methods just couldn't.
The paper then talks about specific types of these depth networks, CNNs and RNNs. Can you explain those?
Sure. So first, convolutional neural networks, CNNs. You might know them from image recognition, like identifying cats in photos.
Right. How does that work for molecules? They aren't pictures.
Well, they kind of can be in 3D. CNNs excel at finding spatial patterns in grid-like data. You can represent a molecule or a protein structure as a 3D grid. A 3D CNN can then scan this structure to find important shapes or features.
Like finding the specific spot on a protein where a drug needs to attach.
Exactly. The source gives that specific example, predicting the protein binding site. It also mentions using 3D CNNs to predict protein ligand absolute binding affinity, basically, how strongly a drug molecule will stick to its target, a super important factor for drug effectiveness.
Okay, so CNNs are good for analyzing 3D structures. What about RNNs?
Recurrent neural networks, RNNs, are designed for sequential data where the order matters. Think of language, the order of words creates meaning.
Or DNA sequences or protein sequences.
Perfect examples. RNNs have a kind of memory, allowing them to process information step by step while remembering what came before. The source mentions using them for tasks like generating focused molecule libraries, creating variations of known drug structures, and crucially, they're also used for something called de novo drug design.
De novo, that means from scratch, right? Designing totally new molecules.
Exactly that.
Yep.
Not just tweaking existing drugs, but inventing completely new ones based on desired properties. And the key tools for this, mentioned in the source, are generative models.
AI that actually creates new molecules. That sounds like science fiction.
It's happening now. These models learn the underlying rules of chemistry and molecular structure from vast data sets. Then, they can generate novel structures that are chemically valid and, hopefully, have the potential to be effective drugs. The paper mentions a few types. GANs, VAEs, and notes, RNNs, can be generative too.
The paper talks a lot about GANs. What's the adversarial part about? How does that help create molecules?
Okay, generative adversarial networks, DNNs, are fascinating. It's basically two neural networks in competition. You have a generator network trying to create realistic-looking molecules, and you have a discriminator network trying to tell the difference between a real molecule from the training data and a fake one made by the generator.
Like a forger and a detective.
Exactly. They train together. The generator gets better at fooling the discriminator, and the discriminator gets better at catching fakes. This constant battle pushes the generator to produce increasingly plausible and novel molecular structures.
Wow. So they use GANs to generate new drug candidates.
Yes, for generating focused libraries, creating totally new structures for specific diseases like cancer, predicting molecules with certain desired properties, even for creating extra training data when real data is scarce. They're very versatile.
What about VAEs, briefly?
Variational autoencoders, VAEs, are another generative type. The source mentions them for dimensionality reduction, simplifying complex data, and then generating new molecules within that simplified representation. Also used for de novo design.
So deep learning isn't just one tool. It's a whole toolbox. CNNs for structures, RNNs for sequences, GANs and VAEs for actually inventing new molecules. It's being applied everywhere in the drug discovery pipeline.
Absolutely everywhere. The review lists tons of other applications. Predicting pharmacological properties, how a drug acts in the body, predicting physical properties like solubility in water, critical for making a pill.
Predicting if it's toxic.
Yes, predicting toxicity. They even mention a specific tool called Deep Tox. Predicting liver injury risk, predicting drug-target interactions, even predicting chemical reactions and helping chemists figure out how to actually synthesize the molecule in the lab. That's called retrosynthesis.
This really paints a picture of AI as this fundamental engine accelerating almost every step. For you listening, the takeaway is that AI, especially deep learning, is shifting drug discovery from slow, expensive trial and error
towards a faster, data-driven computational process, exploring possibilities impossible to reach manually, predicting outcomes, inventing new structures. It's potentially revolutionary for speed and cost.
But the AI needs data, right? And inspiration. Where do the actual ideas for these complex drug molecules come from? Especially the really effective ones.
That's where nature comes back into the picture. Billions of years of evolution. Which brings us to natural products and how we're accessing them now using things like environmental DNA.
Right, back to nature's medicine cabinet. Natural products have a long history.
A very long and successful history. The source calls them a fertile source of therapeutics, especially from microbes. And that statistic is just mind-blowing.
But what about clinical drugs?
Yeah, More than half of clinical drugs from 1981 to 2014 are derived from or inspired by natural products. More than half.
That's huge. Why are they so good?
Because evolution has already done a lot of the work. The source explains they've been evolutionarily selected for biological interactions. Microbes make these compounds to survive, compete, communicate.
So they're already sort of primed to interact with biological systems.
Exactly. They often have these really complex, unique chemical structures that are just more likely to do something biologically useful compared to random synthetic molecules. Nature is an amazing chemist.
But wait, the source also said pharma companies started backing away from natural products in the 90s? Why, if they were so great?
Good question. It was the limitation of the old methods. Back then, it was top-down screening. You'd grow microbes, test extracts for activity, then try to isolate the active compound.
Laborious.
Very. And by the 90s, they kept finding the same things over and over. Rediscovery rates were high, making it inefficient. The easy pickings seemed gone.
Until genomics changed the game.
Totally changed it. Cheaper, faster DNA sequencing meant scientists could look directly at the genetic instructions, not just the final chemical product. This led to huge databases of microbial genomes and metagenomes.
And looking at the DNA revealed something unexpected.
Something massive. They found that these genomes are packed with biosynthetic gene clusters, or BGCs. These BGCs contain the blueprint for making natural products. And the key finding was there are vastly more predicted BGCs out there than the number of natural products we've actually discovered and characterized. Huge untapped potential.
So BGCs, remind us again, they're like the instruction manuals for making these natural chemicals.
Precisely. A BGC is a group of genes, usually clustered together on the DNA, that encode all the enzymes needed to assemble a specific natural product, step by step. Finding a BGC is like finding the factory blueprint. Genomics revealed countless unknown blueprints.
A massive genetic cookbook full of undiscovered recipes. How do scientists find these BGC recipes and all that DNA data?
That's called genome mining. It uses computational tools, bioinformatics, to scan DNA sequences and identify the characteristic features of BGCs. The source mentions tools like anti-SEM-HH. It can find potential BGCs and even predict the basic structure of the molecule they might create.
Predicting the molecule just from the DNA sequence.
Predicting the core structure, yeah. It's powerful. And there are efforts like the MBig database to standardize how BGCs are recorded and shared, helping the whole field move faster.
Okay, finding the blueprints is one thing. But what's the catch? It can't be that easy.
It's definitely not. A major hurdle highlighted in the source is that many of these BGCs are silent.
Silent, meaning the microbe has the blueprint but doesn't actually build the factory or run it under lab conditions.
Exactly. You find the genes, you grow the bug in the lab, but it just doesn't make the compound or makes tiny amounts. The genes are there, but inactive or silent in that artificial lab environment. It's a huge frustration.
Finding treasure maps, but the treasure chest is locked tight. Are there ways to wake up these silent BGCs?
People are trying all sorts of things. The source mentions strategies like activation, tweaking the microbe's own metabolism, engineering the BGC genes directly, trying different host organisms, growing microbes together, co-culture, or adding specific chemicals called elicitors to trigger production. It's a big focus.
And getting access to microbes that you can't even grow in the lab, that's where eDNA comes in.
Absolutely crucial. Environmental DNA, eDNA, lets us access the genetic material of the vast majority of microbes that we simply cannot culture using current lab methods. This is the so-called microbial dark matter.
Microbial dark matter. Sounds mysterious. So you skip trying to grow them and just sequence the DNA directly from, say, a soil sample.
That's the core idea. You extract DNA directly from soil, water, sediment, whatever. That sample contains DNA from everything living there. Sequencing this mix bypasses the need for culturing and gives us a window into the genomes and the BGCs of these previously hidden organisms.
And this allows for metagenome-driven drug discovery, using that environmental DNA mix.
Yes, the source describes a few ways. Older methods involve making metagenomic libraries, cloning chunks of eDNA into lab microbes, and screening them.
Putting bits of the environmental code into easy-to-grow bugs.
Right. But a more sophisticated approach now is targeted sequence-based metagenomics. Instead of random cloning, you look for specific, short DNA sequence tags within the eDNA. These tags are from conserved parts of genes known to be involved in making natural products' key enzyme domains.
Like searching for specific keywords that indicate a recipe is present.
Kind of, yeah. These tags act like fingerprints for different types of BGCs. You use tools like NapDOS or eSnapED to analyze these tags, compare them to known BGCs, and get a profile of the biosynthetic potential in that environment. The real power here is finding novel variants of known important BGC types.
So not just brand new recipes, but exciting variations on classic dishes.
Good analogy. Finding a new twist on a glycopeptide antibiotic BGC, for example. The source mentions finding new congeners, slightly different versions of known drug classes this way. It's great for expanding chemical diversity.
What about just sequencing everything in the sample? Shotgun.
Shotgun metagenomics sequence it all. This can uncover BGCs you weren't specifically targeting, maybe from symbiotic microbes that produce important compounds like ET743. The big challenge historically was assembly.
Putting the shredded pieces of DNA back together from thousands of different organisms sounds nightmarish.
It was.
Yeah.
Especially with short-read sequencing. But the source points to the promise of new long-read sequencing. Getting much longer DNA pieces makes assembly vastly easier, potentially allowing assembly of entire BGCs or even whole genomes directly from the mix.
That's a game changer for assembly. And what if the microbe you're interested in is super rare in the sample?
Then you might use single-cell genome sequencing. Isolate individual cells, even rare ones, and sequence the genome from just that one cell. This lets you get genomes from uncultured organisms that might be missed otherwise. The source mentions the Entothionella example unculturable, but single-cell genomics showed it had a huge, unique set of BGCs.
So eDNA combined with these sequencing techs is really opening the floodgates to that microbial dark matter.
It really is.
Yeah.
Accessing a whole new world of chemical blueprints. And it doesn't stop there. The source also talks about using genetic engineering to diversify these natural product structures.
Meaning taking nature's recipe and tweaking it ourselves.
Exactly. Guided by comparing different BGCs, you can engineer the genes, change the starting materials the microbe uses, modify the main assembly enzymes, or use promiscuous tailoring enzymes.
Promiscuous enzymes.
Enzymes that aren't too picky. They can add chemical decorations, sugars, sulfates to various molecules late in the process. The source mentions using eDNA-derived sulfotransferases to add sulfates to glycopeptides, creating new versions.
And you can mix this with chemistry, too.
Yeah, things like mutasynthesis or semisynthesis. It's all about using nature's starting point and then applying genetic or chemical tools to make novel variations with potentially better drug properties.
Incredible. So for you listening, the big picture is nature is the master chemist, especially microbes. Genomics and eDNA are giving us the keys to unlock this vast, hidden microbial dark matter pharmacy. We can find the blueprints, BGCs, and even engineer them to create new drug candidates. But it's a frontier, and frontiers always have challenges.
Big challenges. Which brings us right to the cutting edge where all this potential meets the real-world roadblocks and what the future might hold.
Right. We've seen how companies like Basecamp Research are trying to weave together AI's power with these huge, unique biological data sets, partly sourced from exploring biodiversity via genomics and eDNA.
The AI provides the smarts to analyze the complexity and design solutions.
While the biological exploration provides the raw material, the inspiration from nature.
Exactly. But the sources, especially that natural products review, are very clear about the major bottleneck that still exists despite all these amazing advances.
What's the number one thing holding back progress right now?
It's explicitly stated as our ability to link BGCs with their cognate secondary metabolites. Finding the gene cluster, the blueprint, is getting easier. Predicting its function is getting better. But confirming the exact chemical molecule that specific BGC actually produces in the real world, that's still the hard part.
The blueprint to actual product problem.
Precisely. It's complex biochemistry between the gene sequence and the final active molecule.
What other big hurdles did the sources mention?
Several. Activating those silent BGCs is still tough, especially in microbes. We don't have good genetic tools for the non-model organisms.
Getting the recipe to actually work in an unfamiliar kitchen?
Kind of, yeah. And related to that is heterologous expression. Taking a BGC from its original, maybe unculturable source and getting it to work properly in a standard lab host like E. coli or Streptomyces.
Moving the blueprint to a different factory.
Right. It involves tricky cloning, gene optimization, making sure all the parts work together in the new environment. Finding compatible host BGC pairs is key. The source mentions needing more versatile gifted host strains that are good at expressing foreign BGCs.
A universal factory worker bug.
That would be ideal.
Yeah.
Plus, fundamentally, vast swaths of the microbial world, entire phyla, have no cultured representatives. We We need totally new ways to grow or study these major branches of life.
And the cost. Synthesizing a whole BGC from scratch.
Still a barrier.
Yeah.
Even though DNA synthesis is cheaper, making a large, complex, optimized BGC is expensive, limiting how many you can test. And then, even if you get the compound, figuring out its exact 3D structure, structure elucidation, and precisely how it works biologically, mode of action determination, remains a bottleneck. Sophisticated chemistry and biology needed there.
So the experimental side, actually making and understanding the molecule, is still a huge effort, even with AI pointing the way.
Absolutely.
Yeah.
It requires advances across the board. But progress is being made. Analytical tools like LCMS and NMR are constantly improving for structure determination.
And screening methods.
Getting smarter, too.
Yeah.
Multi-parametric screens give richer bioactivity fingerprints, helping prioritize compounds and hinting at how they work earlier on.
And clearly, collaboration is key here, sharing data.
Hugely important. Integrating databases, standardizing things like BGC annotations with Maybig, building shared repositories. The source stresses this is essential to avoid duplicating effort and to really crack that genes-to-chemicals code. The more connected data we have, the better the AI gets, the faster everyone moves.
So, looking ahead, what's the ultimate vision? Where could this all lead?
The long-term dream, based on the sources, is that with better sequencing, long read, single cell, smarter PGC classification, new DNA assembly methods, advanced genome engineering, and those integrated data sets, we move towards truly systematic targeted discovery.
And the really ambitious goal.
That eventually, maybe, we could reliably deduce compound pharmacological profiles from analysis of BGCs.
Whoa, so just read the DNA blueprint and predict this will likely be an anti-cancer drug targeting pathway X or an antibiotic effective against resistant bug Y.
That's the potential holy grail. Imagine how that would accelerate things, prioritizing the very best leads before even starting the complex lab work. It's incredibly exciting. For you listening, seeing this interplay, AI, genomics, eDNA, synthetic biology, helps understand why drug discovery is changing so fast. It offers real hope for tough problems like antibiotic resistance or finding new ways to treat complex diseases.
This has been a fascinating journey into a rapidly evolving scientific frontier. We've seen the ambition of companies like Basecamp Research harnessing unique data and AI. We've unpacked the deep learning tools driving drug design. We've explored the vast chemical library hidden in nature's microbial dark matter, unlocked by genomics and eDNA.
And we've also grounded ourselves in the very real challenges scientists face in translating that genetic potential into actual medicines, especially that tough link between gene cluster and chemical product.
It really highlights the power of the new tools, but also the enduring complexity and wonder of the biological world itself.
Absolutely. Opportunity and puzzles abound.
So, thinking about all that untapped potential, especially in the trillions of uncultured microbes out there, here's a final thought to chew on. What other revolutionary medicines might be hiding in plain sight within that biological dark matter, just waiting for this fusion of AI and deep biological exploration to finally bring them into the light? What cures are encoded in DNA we haven't even learned to read properly yet? Thank you for joining us on BioBits and Bytes.